introduce Nat Lenzo now, who's also a nuclear physician at Oceanic and Fiona Stanley Hospital as well, and the Wexford Centre.
Thanks for asking me to talk. I'm not going to be talking about anything political like Harvey. I'm going to talk a little bit about science and about a new trial that is about to start in Western Australia. The new agent is lutetium-177 which is probably going to be called Satoreotide, though we're not entirely sure, from Ipsen. The people that are listed there are chief and associate investigators, and this is being done through Theranostics based at Hollywood Private Hospital. So those are my disclosures. And the times they are changing. I think you've heard about some of this. In 2005, we were using indium octreotide imaging, which was a very poor agent. We had started using things like octreotide, somatostatin receptor agonist treatment, lanreotide and so forth. And then come a decade and all of a sudden gallium octreotide is state-of-the-art imaging for neuroendocrine tumours, though it's not Medicare funded. So it's the best test we have for imaging neuroendocrine tumours, and it's not funded by Medicare. I suppose that is a bit political. Lutetium-177 octreotide is also probably the most efficacious treatment we've seen, but we need to get a bit more data, and that data is coming. In 2020, we may not be using either of those two agents, and I'll just show you some of the data that is starting to come about. So there is a little bit of science here, and I'm going to try and do it nice and slowly. And basically, there is, when we talk about somatostatin receptors, everyone knows somatostatin receptors, yay? No? They're on the surface of neuroendocrine tumours. That's what we target with our therapy. That's what we target with our imaging. So that we have a protein which binds to that receptor, which is like a molecular signature on the tumour, and then we can image that with gallium octreotide, or we can treat that with lutetium octreotide. So we've been targeting these somatostatin receptors, but we've been targeting somatostatin receptors with an agonist, which I'll go to in a second. And it was always thought that that was much better. It was much better because it would bind to this activated receptor on the tumour, then the tumour would bring that into the cell, so it's enclosed in the cell. The gallium isn't going to escape anywhere, the lutetium isn't going to escape anywhere, that's nice and safe. So that was always the theory. But you have far more unactivated receptors on the tumour. So if you see here, the agonist will bind to this activated receptor, but this is an unactivated receptor. And there's at least fourfold, if not many times more, unactivated receptors. So Ginsedel in 2006, he found a new type of protein. He found somatostatin receptor antagonists. They bind to both the activated receptors and the non-activated receptors. So there's many more potential binding sites. And everyone thought, oh yeah, look, we're going to run into trouble now. Because it's not going to be taken into the cell, it's going to leach out and people are going to get sick. It's not a good agent. In fact, that didn't seem to actually eventuate. And in fact, when they started looking at it, first in those awful animal models, they found that in fact, just look at the level of darkness, much darker, much more binding, heat more binding. So they thought, well, this might be interesting. And so the antagonist binds both to activated and unactivated receptors. Octreopharm were a company that developed this. And this is the dreaded mouse. The dreaded mouse, which shows that when we look at gallium octreotide or dotatate, same mouse, much higher uptake with the antagonist. So then they started thinking, well, this might be quite interesting for an actual imaging agent. And in fact, this is some of the original images. And you'll see there's far less background, if you've ever seen any gallium octreotide images, far less splenic activity, and I'll show you a few more, but they've compared it to indium octreotide, gallium octreotide, and this. So you'll see far less splenic activity, much more definition. So there are new studies currently underway, and it may very well be, once this goes through all the different processes, that probably, as I said, in 2020, we may be using this. The one good thing, and I'll talk about that probably towards the end, the one good thing that's happened is Octrea Pharma were bought by Ipsen. Ipsen are a bigger company, got more money to spend on this. Octrea Pharma are a small company. But Ipsen have been quite smart. In the past, when people made octreotide, they gave it out to everyone. So Harvey was playing with it, and Peter Mac were playing with it. People were using it. What these companies will do is they've got the money to do the proper trials that will actually convince government. Because it's in their interests. Because they want to get it on the PBS. And that's in your interests. So there are two ways of looking at how you play this game, and it is a bit of a game. But I think that approach will probably get things to market quicker because they have the money, and will probably help more people in the long term. This is the lutetium agent. They call it, for some reason, they call it OPS-201, whereas the imaging agent they call OPS-202. There must be some slight change in the molecular structure. And this is same patient, same tumour, comparing OPS-201 to lutate. So this is the lutetium octreotide, the therapy, published in the Journal of Nuclear Medicine, showing that it seemed to have better activity. They've done some really fancy stuff, and once again, it's about 3 to 4 times increased dose to the tumour. So it may very well be that this will be more effective. But you've also got to remember that may be 3 to 4 more times increased toxicity. Some more images. This is gallium dotatate after treatment with the same agent, with lutetium OPS-201. So it does seem to work, but these is, as David said, 1 or 2 cases don't make, well, a swallow doesn't make a summer. So these are just case series. However, in saying that, there are a whole series of new agent trials. So Ipsen have actually commenced a trial in the US comparing indium octreotide, because you've got to remember gallium octreotide is not available in the United States. Indium octreotide is, so they're going to do a comparison trial, and it will be far superior to indium octreotide, and it will probably be listed by the FDA. In New York, they're currently doing an early phase lutetium OPS-201 trial in neuroendocrine tumours, and that's really a dose findings trial. They're trying to work out, well, what's the toxicity? If this is 3 to 4 times, 5 times more potent, do we use 7.4 gigabecquerels like we currently use? Do we use 5 gigabecquerels? Do we use 2 gigabecquerels? So that's what they're trying to work out at the moment. Going on from that, we've been involved in helping develop the actual protocol for the new phase 2, phase 3 trial, which is 7 sites around the world. So Hollywood Hospital and the Peter MacCallum are the 2 sites in Australia. There's 1 site in the UK, 1 site in New York, and there's 4 sites in Europe. It's already gone through our ethics at Hollywood Hospital, but we're still waiting on the final data from New York on the dose, exactly what dose we're going to do, and then we will start recruiting. So we were hoping that was all going to be ready by now, but like most of these trials, they take longer than you think. So it looks as though it'll be October, November before we actually start recruiting. The parameters are very similar to what gets you into the control nets. You've got to have a metastatic JEPnet, low Ki-67. You can't have any previous lutetium therapy, no significant renal disease. It's got to be progressive disease on imaging. This is lutetium therapy alone. This is not combined. So it's different to the control nets where there is a combined arm. Everyone's got to have an initial gallium octreotide scan, though I think in the US they still allow indium octreotide. There's only 45 patients going to be recruited on this trial, so it's not a very big trial. But there's a lot of follow-up. There's a lot of imaging, a lot of blood tests, a lot of clinic visits. They're fairly pedantic. Mostly Germans have written the protocol. So it's a very Germanic protocol, but it will basically show whether this is safe and effective. And then time will tell whether there will be a comparison trial versus lutetium octreotide. There may not be, because if this gets funded, this gets presented to government, this may, if it's effective, be what gets taken to the PBS. So if you're interested in this trial, those are some numbers, and you can contact us. As I said, recruitment will start, we believe, October, November. And that's it.